So imagine you've got two totally distinct, fully functioning cities, like complete with their own complex infrastructure and, bustling citizens, interconnected economies. And they're just suddenly smashed together overnight.
Oh, man.
Right. Not in a destructive action movie kind of way, but just a scenario where they suddenly have to coexist
Yeah.
in the exact same physical space. Like the subway lines merge without warning, the power grids cross over one another,
Yeah, total chaos.
and the citizens of both cities are suddenly walking down the same crowded streets.
Yeah.
You immediately have to wonder, like, who takes over the local government? Whose economy becomes the dominant one?
Right, right. Do they blend into a hybrid, or does one just bulldoze the other?
Exactly. Does one just completely take over the identity of the other?
Well, it's a chaotic image, but it actually perfectly captures the sheer scale of what happens every single day at a microscopic level. It's this ecological phenomenon called microbial community coalescence, or MCC. Because, we tend to think of microbes in isolation, like growing in a petri dish. But in reality, they exist in these massive, hyper-complex ecosystems, and those ecosystems are just constantly colliding.
Right, like with flooding.
Exactly.
Yeah.
Think about the flooding of a coastal area where highly adapted marine microbes suddenly wash into a freshwater basin. Or massive agricultural operations where they dump tons of bioorganic soil amendments onto an established field.
Wow.
It even happens during something as simple as intimate human kissing.
Oh, yeah.
You're passing an entire microscopic ecosystem from one mouth to another. And crucially for modern medicine, it's the fundamental mechanism behind treatments like fecal microbiota transplants. We are just constantly mashing these invisible cities together, and the biological fallout is immense.
Okay, so for this deep dive, our mission is to unpack this really groundbreaking 2025 paper from the ISME journal. It's titled Interplay of Ecological Processes Modulates Microbial Community Reassembly Following Coalescence.
Which is a very academic way of saying we are finally getting a look at the mathematical mechanics of what happens when these worlds collide.
Right, exactly. The researchers are trying to disentangle two massive opposing forces that basically dictate the outcome of these collisions. On one side, you have abiotic filtering, so the physical and chemical constraints of the environment itself.
The infrastructure of our city analogy.
Yeah, the weather patterns, the layout of the streets. And on the other side, you have biotic filtering, the actual fierce microscopic warfare and cooperation between the individual species themselves.
Right, and disentangling those two forces has been kind of a holy grail in microbial genomics. Because historically, when researchers wanted to see which microbes actually survived a merger, they relied on tracking tools that look for specific individual genetic sequences.
Like Source Tracker.
Yeah, Source Tracker or FEAST. They were built to follow a specific strain from the donor community to see if it showed up in the final mixture. But the problem is, as biological diversity increases in these ecosystems, those traditional tools hit a massive wall.
Because of functional redundancy.
Exactly.
Okay, let's unpack this. Because functional redundancy is such a critical roadblock in genomics. We aren't just talking about a backup system. We're talking about totally different species evolving the exact same metabolic machinery.
That is the core issue right there. So imagine you're tracking one highly specific strain of bacteria because you know it digests a certain complex carbon.
Okay.
If that specific strain dies out during the violent merger of these two communities, the old tracking tools might tell you, oh, the community lost its carbon digesting function.
But it actually didn't.
Right, it didn't, because in the soil, 3 totally different taxonomic strains might have immediately stepped up to consume that carbon using the exact same chemical pathway.
Wow.
The function is completely redundant across the ecosystem, so looking for individual specific strains totally misses the forest for the trees. It creates this incredibly noisy data set where a community looks mathematically chaotic, even though its overall function hasn't changed at all.
So if tracking individual genetic sequences is giving us a flawed picture of the merger, how do you actually measure the resemblance between a massive donor community and the final mixed outcome? Because this new statistical framework, it clearly bypasses the old genetic tracking methods entirely, which I find brilliant.
Yeah, it's a totally different approach.
They zoom out and essentially ask, does this new haystack look more like haystack A or haystack B?
Exactly. They built a beta diversity-based statistical framework, so instead of asking if microbe X survived, they quantify the relative contribution of the entire donor community to the newly mixed outcome community.
Right.
They take these massive count tables representing the taxonomic profiles of the microbes, and then they calculate the mathematical distance between the whole original community and the new one. And they rely heavily on a metric called the Jensen-Shannon divergence, or JSD.
Okay, let's translate JSD into something we can visualize, because throwing around statistical divergence metrics can get abstract really fast.
Oh, for sure.
Imagine you have two incredibly complex recipes for a massive pot of stew.
Right,
JSD doesn't just check if both stews have carrots in them.
right.
It mathematically measures the exact overlapping ratio of every single ingredient. The salt, the broth, the potatoes, the spices.
Yeah.
To tell us precisely how much of Recipe A's core identity survived in the final merged pot.
That's a great way to put it. And a smaller JSD distance means a tighter mathematical overlap.
Got it. It's essentially a resemblance test for the entire ecosystem.
Yeah, and that resemblance test gives us a way to track the collision over time. That time series aspect is critical here. The researchers mapped this out on day 1, day 5, day 15, and day 30.
Because these microscopic mergers are highly volatile.
Highly volatile. A donor community that looks completely dominant on day 1 might collapse entirely by day 15 once the easily accessible nutrients are depleted and the biological warfare really kicks in.
Right, so to put this framework to the test, the researchers designed a really clever microcosm experiment. They took 3 highly distinct natural soils from Delaware, Michigan, and Nebraska. Let's just call them soils A, B, and C.
Okay.
And they mixed them in various 1-to-1 ratios.
Hmm.
But it is vital to point out the chemical reality of soil C here. It was a massive outlier.
Oh, yeah.
It has significantly higher clay content, higher organic matter, and a much more alkaline pH compared to the others.
It acts as an incredibly heavy thumb on the scale. The physical chemistry of soil C is unforgiving if you aren't evolved to handle it.
But before they forced these soils to merge, they did something that I really have to push back on initially.
Okay, what's that?
They manipulated the biological complexity of the source communities using a method called dilution to extinction.
Right.
They created serial dilutions of the microbes, taking them down by factors of 10, all the way to 10 to the minus 5.
Yeah.
If the ultimate goal is to study how complex communities behave when they mix, why water them down first?
It seems counterintuitive.
It sounds like you're randomly deleting lines of code in a software program just to see when the whole system crashes. Like, why intentionally destroy the very ecosystem you're trying to study?
Well, what's fascinating here is that the destruction isn't a flaw in the design. It's the entire point of the experiment.
Really?
Yeah, because in microbial ecology, a highly functioning community isn't just a random bucket of single-cell organisms. It's an intricate, delicate web of metabolic interdependencies.
Okay.
Like microbe X might produce a highly toxic waste product that microbe Y actually needs to breathe,
Oh, wow.
while microbe Y produces an extracellular enzyme that microbe Z requires to break down its food. This is known as ecological cohesiveness. They are locked together in a state of syntropy.
They're essentially running off each other's chemical exhaust.
Exactly. So when you perform a serial dilution down to 10 to the minus 5, you aren't just lowering the overall head count in the dirt. You are forcing a stochastic, completely random disruption of those vital ecological interactions.
You're breaking up that cohesiveness.
You're breaking it up. The rare species are washed out entirely. The metabolic assembly lines are severed, and the community's structural integrity is compromised.
Oh, okay. So by deleting that code, they isolate the variable of biological complexity itself.
Exactly.
They're asking whether a fragmented, chaotic community performs differently during a collision than a fully cohesive one.
It gives them a sliding scale of biological strength to test against the physical environment. It sets up the ultimate tug of war.
Right. And when they finally smashed these fragmented microscopic cities together, combining soil A with soil B, A with C, and B with C, the data revealed exactly how powerful the physical environment really is.
Oh, it was absolute.
Yeah, let's follow that collision. Because the dominance of soil C was just absolute. When the researchers mixed soil C with either soil A or soil B, soil C completely took over.
It wasn't a delicate blending of two cities at all.
No, it was a total unmitigated takeover by the microbes native to C.
And this is a pure demonstration of home field advantage driven by abiotic filtering. Soil C imposed a stringent, unforgiving environmental filter on the incoming microbes. The environmental properties, specifically that high pH and its massive nitrogen pools like ammonium and nitrate, they dictated the terms of survival on a cellular level.
Right.
Because changes in pH fundamentally alter how proteins fold and how microbes generate cellular energy. If an incoming microbe from soil A wasn't pre-adapted to that specific alkaline environment and those nitrogen levels, it didn't matter how fierce a competitor it was back home. It simply couldn't function.
And the numbers attached to that takeover are staggering. The statistical framework showed that those abiotic factors explain more than 90% of the variance in the community structure over time.
Which is huge.
Over 90% of the outcome was decided purely by the chemical infrastructure of the dirt.
Yeah, donor seed dominated from day 1 all the way to day 30 across all those different dilution levels we just talked about.
Even the highly diluted ones.
Exactly. Even if they took soil C and highly diluted it, artificially breaking down its ecological cohesiveness, the microbes native to C still won the merger. The abiotic environment of soil C was so extreme relative to A and B that the physical environment simply bulldozed the biotic interactions. The environment was the only thing that mattered.
Here's where it gets really interesting, though. What happens to the math when you remove that dominant physical environment from the equation entirely?
Right, the A and B mix.
Yeah, when they forced soil A and soil B to merge, the dynamic flipped completely.
Because soil A and soil B shared much more similar physical and chemical properties, neither soil imposed that crushing abiotic filter. The infrastructure of the two cities was effectively a tie.
Right.
And in that vacuum, without a dominant environment dictating the rules of survival, the microbes themselves had to fight it out.
And the environmental shifts in the A versus B mixtures explained less than 45% of the community turnover.
Oh.
So the physical environment took a massive backseat. And interspecific interactions like competition for trace minerals, chemical warfare via antibiotics, and cooperative metabolic sharing became the primary engines driving community assembly.
And this is exactly where the dilution to extinction setup really proves its worth. In the A versus B mixture, the dilution level dictated the entire timeline of the collision.
How so?
Well, at the lowest dilution, where the two communities were fully intact, highly diverse, and cohesive, they fought to something of a symmetric stalemate by day 30. They integrated.
Okay.
But at the highest dilution, 10 to the minus 5, where those vital species interactions had been randomly ripped apart prior to mixing, the outcome was total ecological chaos.
Right. The timeline data for those diluted mixtures looks like a massive pendulum swinging back and forth.
It's ecological drift on full display. Because the stabilizing interactions were gone, there was no biological anchor.
Yeah.
Donor A's fragmented community might surge and take the lead on day 5, only to completely collapse by day 15.
Letting donor B take over.
Exactly. It conclusively proves that in the absence of a strong environmental filter, the internal complexity and the interconnectedness of a microbial community is the only thing that determines its ability to successfully establish itself in a new space.
But structural dominance, winning the genetic battle and taking over the physical space, is only half the story in genomics.
Yeah.
We have to ask, does winning the biological war actually mean they control the economy of the dirt?
Right, the function.
Yeah. How did the researchers prove that this shifting community structure actually changed what the soil was doing?
To connect the structural data to real-world function, the researchers had to track the community's metabolism. They used these biolog ecoplates to measure how aggressively and efficiently the newly formed communities could digest 31 distinct types of carbon.
Okay.
They also measured extracellular enzymatic activities, essentially tracking the specific chemical tools the microbes were pumping out into the environment to break down complex nutrients.
So they're measuring form versus function. And the results here perfectly highlight why our earlier discussion about functional redundancy matters so much to applied science.
Absolutely. In the highly diluted samples where the researchers had artificially stripped away the microbial diversity, they found a direct, unbreakable, positive correlation between the community's structure and its function.
Because there's no backup team.
Precisely.
If the specific bacteria responsible for breaking down a complex sugar fails to survive the merger, that sugar just sits there. The function is entirely lost.
Because the dilution process removed the functional redundancy, the failure of specific specialized taxa to engraft during the coalescence directly changed the entire community's metabolic output. But in the low dilution samples, the highly diverse, fully intact communities, the structure and the function entirely decoupled.
So even if a massive wave of specific strains from donor A lost the biotic battle and died off, the overall metabolic function of the soil barely flinched.
Right. In those rich, diverse mixtures, there were always multiple other species from the opposing donor community ready to step into that ecological niche and perform the exact same metabolic job.
The players on the field changed entirely, but the score remained the same.
Exactly.
So what does this all mean? It's easy to get lost in the soil chemistry and the dilution metrics, but this beta diversity framework solves a very human problem in microbial genomics.
Oh, if we connect this to the bigger picture, this macro-level approach is vastly superior for predicting functional outcomes in hypercomplex biological systems.
Right,
Take fecal microbiota transplants, or FMTs, in modern medicine.
right.
We use these to treat incredibly severe, life-threatening gut infections like C. diff.
Mm.
We take a healthy, diverse microbial city and parachute it into a sick patient's gut, hoping it will outcompete the pathogenic bacteria.
It's the ultimate high-stakes microbial coalescence event.
It really is.
But we've historically tried to predict if an FMT will cure a patient by tracking if a handful of specific probiotic strains survive the journey.
Which this paper proves is an inherently flawed approach.
Right, because it completely ignores functional redundancy.
And more importantly, it ignores the abiotic environment of the patient's gut.
Hmm.
The inflammation levels, the mucosal pH, the specific nutrient availability caused by their diet.
Wow. Yeah.
This framework argues that to predict the success of a microbial therapy, we need to stop obsessing over individual strains. We need to measure the overarching structural and functional resemblance of the whole community using tools like the Jensen-Shannon divergence.
Predicting engraftment based on community emergent properties.
Exactly, like overarching resilience and resource utilization.
Right.
That is the absolute future of microbiome therapies. You aren't just dropping in a few helpful bugs, you are dropping in an interconnected ecosystem, and you need to measure it like one.
It's a massive paradigm shift. It forces us to stop staring at the genetic sequence of a single probiotic strain and start looking at the battlefield it's being deployed into. Yeah. This framework cuts through the extreme noise of microbial complexity and gives researchers a hard mathematical way to definitively say, oh, okay, the chemical environment is driving this merger, or the biological species interactions are driving this merger.
And if we follow that logic, it leads to a rather unsettling implication about how we live today.
What do you mean?
Well, we saw that in the presence of a strong abiotic filter, the physical environment dictated over 90% of the microbial community's success. 90%.
The ultimate home field advantage.
Right. So if we can theoretically engineer environments in agriculture or medicine where the specific microbes we want inevitably win, it makes you wonder about the environments we are accidentally engineering right now.
Oh, wow.
Every time we ingest microplastics or consume heavily processed chemical emulsifiers or drastically alter our soil with synthetic runoff, we aren't just changing our diets or our farming yields. We are radically altering the abiotic infrastructure of our own biology and our planet.
Because if the environment dictates 90% of microscopic survival, we might be unintentionally setting up a massive home field advantage for pathogens.
Exactly. Pathogens and metabolic dysfunctions we haven't even fully recognized yet. We are changing the city infrastructure. We have no idea which citizens are getting ready to move in.